was probably the first thing that came out because of the guy who had the interaction with the lady in the park, the
Okay.
bird watcher. I think that was the first one. And then I saw it kind of take off too, like after that with all of these opportunities. So
And so, as a student, if you are interested in something in particular, there might be a group out there of people who resemble you or have a similar type of knowledge of the culture as you, and you can ask them about that particular profession. Is that something that you actually want to do? I would take advantage and use their insight as a way to figure out if this is a profession that you actually would want to do. In addition, if it is, they're doing it already. And so you can make contact with them to figure out if there are any internship opportunities or post undergraduate opportunities. So that means careers or graduate school or master's schools. We want you to come here for master's school. But if you end up going somewhere else, we're still excited for you also. See, that was my little master's of public health plug. There you go. All right. I'm going to stop being silly and I'm going to present to you today. I usually introduce the speaker. So I'll try to introduce myself. I actually went to Virginia Union as an undergraduate and graduated 1999. So I just dated myself. Well, last century. Feels like last century. So I have seen old stuff and I'm still current or trying to be current. And so I came here with the intent of exposing students to the stuff that I had seen related to neuroscience and the different techniques that I've seen in other labs that you might not necessarily see unless you end up in that particular lab or that particular institution. But there are a lot of common techniques and schools of thought that need to be adapted by just the researcher in general. And so I came back here and I have picked up a nice group of students, young researchers to work with, and we're exploring some of the topics that I have been investigating. So I'm going to pull up my PowerPoint now and show you what's been going on. And the main topic today that we will be discussing is the regulation of intracellular in the mammalian brain. So can everybody see my slideshow?
Yes.
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 Okay. If you ran into me within the last 4 or 5 years here, you did not have to do much to get me to start talking about this. I have taken this topic, which is central to all of cell physiology and biology just in general, and tried to infuse it into the curriculum so that students understand how important pH is in controlling or regulating or modulating the function of the cell. I, in particular, focus on the brain. And so, what does the brain do? It does just about everything. I mean, it's the reason that you see, it's the reason that you hear, that you speak. It's even why you can move. It gives you the consciousness, it controls your unconsciousness. It does the reflexes in response to negative stimuli. It also is the reason that you're attracted to certain things. And so the brain is taking all of this information that is outside and inside of you and interpreting it to give you the reality that you know. And that is why you have your identity and why you're able to think the thing that makes you unique. The brain is basically controlling that. And we want the brain to do that. As we develop, we start saying new things and we learn about those things and actually start applying it to our own lives. And these things, that's what learning is, right? And so, what can happen to the brain as a result of not functioning right? Well, I'm going to be focusing on acidosis, which is the measurement. Well, pH is the measurement of the level of acid that is in a particular solution. And acidosis can be both metabolic which means there's chemical reactions that are taking place and acid is the byproduct, or which means that cellular metabolism that's taking place, the byproduct of that, which is carbon dioxide, if it's not properly expelled, it can also cause some type of acidosis. So this is just the outline I'm going through. And you're going to understand after I give a brief foundational understanding of protein, how they regulate cell function, and how acid regulates actually the protein's function. So if proteins regulate cell function and acid regulates proteins, then indirectly acid influences cell function. And so how does the cell deal with this particular acid that's present, whether it's metabolic or respiratory? Well, there's special proteins that actually regulate intracellular pH. And as I said, acid affects protein, so acid actually affects the proteins that regulate pH. So I'll talk about the different techniques that I'm using. This is an older presentation, so there's new stuff that has come out, but I'm not going to present it until it's actually published. So it should be published in a couple of months, but it's very, very interesting. But this is the foundation of how I was able to get the results that are going to be coming out. I'm using this different technique called fluorescent recording, where I'm recording the intensity or the emission of a particular dye that has been stimulated. This dye is BCECF, and it's a measurement of intracellular pH. And I'm using this ammonium pre-post technique to intentionally acidify a cell and then watch it recover from the acidosis. In that way, I can actually measure the steady state intracellular pH, and that's what this is, or the amount of acid inside of a cell during steady state. And I'm going to be looking in the rat hippocampus. The hippocampus is the area of the brain that controls memory and learning, like some of the stuff I was just describing with the functions of the brain. And then I'm going to look at the recovery from this acidosis, which would be a model for this metabolic acidosis or this respiratory acidosis that we just talked about, the brain when it's dysfunction actually having. And this can be realized clinically in a bunch of different diseases. So you've heard of Alzheimer's, and you've heard of even concussions or tumors or multiple sclerosis, but this particular study is not only focused on when the brain goes wrong, and that would be these different diseases here, but when the brain is just normally functioning, you're constantly making acid. And so what does the brain, or what do the cells in the brain do with this acid to make sure that the acid level doesn't get too far off? So I mentioned metabolic acidosis here, and I mentioned respiratory acidosis. Just as a little background, this right here represents a typical cell, animal cell. And you'll see that you have all these organelles inside of the cell doing different things. And inside of the nucleus, where the DNA is, you can even go even closer and see the nuclear pores. And these are proteins that are in the pores of the nuclear membrane that allow things to come back. You also have the mitochondria here. And even in the mitochondrial membrane, there are proteins there. So proteins are basically everywhere in the cell. They're in the membrane, they're floating around loosely in the cell, and they're doing all of the functions of the cell. And when these proteins actually are impacted by acid, when it comes from a metabolic source, you have metabolic acidosis. What can cause metabolic acidosis? If you have diabetes, you end up with metabolic acidosis. We've heard of that before. If you starve yourself, if you have alcohol, there's an association of alcohol with the production of acid. Even toxins, so methanol, aspirin. You can even just drink acid. All of these different ways will give you a metabolic acidosis. Or if you have severe diarrhea and you're getting rid of the base, well, you have gotten rid of the base that would have balanced the acid. So there's more acid now because of this diarrhea. Or if you end up with like a kidney disease. These are ways that you can get metabolic acidosis. And keep in mind, when we say acidosis, we're talking about the hydrogen ion. We're talking about the hydrogen atom that's lost its electron, and now you just have a proton. So it's an ion. It's a positive charged ion. So how does the body get rid of this acid? Two major ways that the body gets rid of the acid. You have your lungs. So you breathe in oxygen. The oxygen goes around to your cell. It's used for metabolism. And you blow out CO2, the product of the metabolism, comes around to your lungs and you blow that out. So the respiratory system is the way that you can quickly change the amount of acid inside of the blood. The longer or more chronic term way to get rid of acid is to actually use the renal system. So you're urinating out acid. But the brain wants to control what's actually taking place. So as I even mentioned that the lungs are blowing out CO2, it's actually the brain that's controlling the lungs. You have specialized cells on the surface of the brain, and these letters here represent the respiratory areas. You also have cardiovascular areas in close association with these respiratory areas. And these letters here, these Roman numerals, they represent the cranial nerves that are coming out of the brain. You're actually looking at the surface of the brain. So if you feel your trachea or your throat, right behind your throat, that's still your brain as it goes into your spinal cord. And so this part of the brain is actually sensing changes in the amount of acid in the blood. And if the amount of acid in the blood gets too much, these areas, like this C right here or this R right here, send signals to the lungs from the cervical C3 through 5 to drive the diaphragm to cause respiration to take place. The sensor is up here, which drives the cervical nerves to cause the lungs to breathe. If you want to see other areas in the brain that are involved in respiration, previous studies stimulated just that area with small pieces of acid, and they saw throughout the brainstem. This is a sideways view of the brain. If you that area right there is this area right here. So we've turned the brain sideways. If you stimulate right here with acid, several areas in the brain also become activated. So that means that they have to be synaptically connected to these areas. Not all areas, just certain areas. These areas have other functions. For example, this dorsal medial nucleus is involved in digestion. This area right here is involved in, the supraoptic nucleus is involved in water balance. And you have other areas that are involved in like vomiting, the area postrema, but they have a dual function. That function that I just mentioned and respiration, because they're becoming stimulated by a respiratory stimulus. There's also chemical reactions taking place in the body to try to get rid of some of this acid. This acid, or this H plus, combines with bicarbonate to form carbonic acid, which changes into water and carbon dioxide. And that's another way you can blow it out, just like this. But you have to remember that the source of the acid has to be eliminated for the acidosis to be eliminated. And because this ventral area of the brain is involved in all of these functions, whether it be water balance or hunger and eating or vomiting or cardiovascular, they're all on the surface of this brain. And the theory is that this ventral surface is the main switch for all of these different physiological functions. So that's what the research really is driving to. But in order to tackle that, I have to tackle the individual cell and how it actually functions in the face of acidosis. So that's the reason why I'm doing this study. So let's see what type of data I'm getting from that. Just as a review, which you probably don't need, the electrons are spinning around the nucleus, and where the electrons aren't provides room for atoms to combine in different types of chemical bonds. And this is the periodic table. You don't even need all of these elements to be for the to do biology. Like we only have, what is it, of the first 20 really, and all the other ones after that are just specialized for different functions. But the reason that I mentioned that is because remember that in the nucleus you have this DNA taking place, and that DNA turns into RNA. This is the central dogma I'm talking about. The DNA turns into RNA, and the RNA turns into amino acids. And you string amino acids together and you have this protein. Well, we're saying that proteins control cell functions, and we're saying that acidosis controls protein. So right now we're just looking at the different amino acids that can make up a protein. The thing that actually makes amino acids unique is the part that's highlighted in pink. That's the side chain, which is represented here as an R. This is the typical structure of a generic amino acid. And the R, depending on what's in that pink, gives that amino acid different properties. So these ones right here, they're non-polar. They don't like water. These ones are polar. They like water. Keep in mind that non-polar and polar, they're only talking about covalent bonds. These are positively charged and negatively charged. So now you have ions. And then you have ones that actually have shapes, aromatic side chains. And so based on what is in that R position, when the amino acid string starts forming, you have interactions between these amino acids, whether they're positively binding to negatively or attracted to negatively ones, or based on how big the side chain is. You're talking about the folding of the amino acid. It's the shape, the overall shape of this amino acid chain that determines its function. So we want to know the amino acids that make up the protein because their order will determine the shape, and the shape will determine the function. But I said that pH or acidosis can also impact the function. Why? Because acidosis is a positively charged, or hydrogen is a positively charged species, and that will interact with positively charged ones. Positive things and positive things will repel each other. Positive things and negative things will be attracted to each other. So depending on the level of acid in the cell, you can actually change the shape of a protein. But remember, protein's shape determines its function. So that's actually how pH is changing the function of a protein. What are examples? Well, you have ion channels. Proteins can be ion channels. They can be receptors, transporters. They can be pumps. Of the ion channels, these are different types of channels. So you have potassium ones, sodium activated potassium ones. You have acid sensing channels. And the amount of acid does different things. Acidosis decreases these one, it reduces this one, it increases this one. So how do you know exactly what to do with the acid unless you actually understand the structure of the channel? This one right here is actually from 2008, and the data from that I'll be showing later on in this presentation. But you also have receptors. The current gets reduced when acid is present. Transporters, expression of the protein increases, activity increases, and then you have pumps increases. And so these are links to different papers that talk about these particular channels and the effect of acidosis. Well, we're going to look at this one right here, this sodium activated potassium channel. It's called SLAC because it's a sodium-like activated calcium sensing potassium channel. The amount of acid that a cell likes to have is around pH 7, slightly basic 7.2, 7.4, but it's really in this range. So there's really a tiny amount of acid equivalents inside of the cell. Like I said, 7.3. I'm doing a technique called single channel inside out patch clamp technique, which is when you take a small thin electrode like a straw and you press it up against the cell membrane. It's so small that you can isolate single channels and monitor the electricity or the current passing through those channels. So right here in A, you start off at a normal pH. 0 is this line here. And when the channel opens, it's a downward opening. So here's 0. This is an enlargement right here of that area, just to show you what's happening. This is 0. And all of a sudden, a channel opens, closes, opens again, closes, flickers open, opens, and then 3 channels open, 2 of them close, it opens again, 3 channels open, 2 of them are open, 1 of them is open. So you can actually see the channels opening and closing. Do you guys see this? So this is the channel opening, and then here's another one that just flickers open and closes. This is the activity of the channel during rest. When I change the pH from 7.3 to 6.3, the channel stops opening. This enlargement shows that there's a single channel opening. And you know it's the same channel size because you're looking at the level of the opening. If you put a ruler across here, you'll see that all of these channels are opening to the same size. When you take it out of the acidosis and you put it back, the channel activity comes back. So we're actually looking at potassium moving through the cell. And if you remember from neurophysiology, or you remember about the biology of a nerve cell, you need the potassium to leave the cell in order for the cell to repolarize and go back to an unexcited state. If this doesn't happen, the cell becomes excitotoxic and it will die. And this is how ischemia happens, or this is how strokes take place. So we're able to show the normal activity of the cell's channel, the sodium activated potassium channel. And then we put it in acid and we see that we make the channel stop its activity. And then we take it from the acid and put it back and we see that the activity is restored. So that means that when we made an acid, something happened with the channel to prevent potassium from going through it. Maybe the structure of the channel changed to prevent potassium from moving. So here's another experiment. We have it at the normal amount of pH, same size channels opening. And then we take away acid. So we made it more basic. And you see that the activity increased. And then when we put it back in the normal solution, the activity decreases again. So these are just examples of the effect of increased acid, which is acidosis, and decreased acid, which is alkalosis. And if you look at the pH here, and this is a measure of the opening probability of the channel. At this pH, the channel is not really open. As you increase the pH, the channel starts opening more to here, which is the control. And then as you increase the pH, the channel continues to open. So you have a sigmoidal curve here, reflecting the channel's activity. Remember I said that CO2 was respiratory acidosis? Increased CO2? Because here, I mentioned it here, respiratory acidosis. I mentioned how the lungs are necessary because you're blowing out the CO2. And if you don't blow it out, you end up making more acid. So here, I did regular acid. This was metabolic acid. Now I'm looking at respiratory acidosis. So I got to increase the amount of CO2. This is the amount of CO2 in the air that we normally breathe because we're making the CO2 and we're blowing it out. Inside of our bodies, we have around 5% CO2. So I'm going to compare breathing normal air that has no CO2 with adding CO2, which is a form when combined with water becomes acid or dissociates to acid. And you can clearly see that there's a lot of activity here. But then when you put CO2 there, the activity decreases. When you take CO2 away, the activity increases again. This amount of CO2 would be representative of a severe hypercapnia. So I've increased the CO2 amount even more. And it shows that as this is the concentration of CO2. As the concentration of CO2 goes up, the opening probability decreases. So both metabolic acidosis stops this activity and respiratory acidosis stops this activity. So how does the cell prevent this acidosis from taking place? You have these proteins. Remember I mentioned that you have proteins here that, where is it? Well, you have proteins here that regulate the amount of acid in the cell. That would be acid-base transporters that regulate pH. So that's what we're looking at right now. This is one family, and this is another family. What makes them a family? Well, these ones right here, they go through the membrane 12 times. These ones right here, they go through the membrane 14 times. These ones up here, they have a long C terminus. These ones down here, they have a long N terminus. So there's things in common with them. Not only are they in the membrane the same way, but they also transport the same things. These ones exchange extracellular sodium for intracellular hydrogen. So you're actually exchanging sodium and hydrogen. It's a sodium-hydrogen exchanger. So there's more sodium outside, there's hydrogen inside, you're exchanging it, and that's a way to get rid of acid. Then another way is to actually bring in bicarbonate. So this is published in 2014. Some of the people that have interacted with me have been asked to read this paper, and so they should understand that if these acid-base transporters aren't functioning, then you have a problem regulating your pH, and you probably have some clinical disease. How are they actually doing it? Well, we want the pH to be a certain level. So if you think about how the pH level is, you're either making it acid or you're getting rid of acid. And it's the balance between how much you make and how much you get rid of that determines your pH. It would be similar to having the faucet on in the sink. And you don't want the sink to fill up. So what do? You take a cup, and as the sink starts filling up, you take a cup and you pour it out. So you're keeping the level of water in the sink at a certain level. You're keeping the level of acid in the cell at a certain level by either extruding or getting rid of the acid. So as the acid becomes, if this is pH, as it becomes more acidic, you're going to get rid of more. As it becomes more basic, you're going to start loading acid. So it's the balance between the extrusion rate and the loading rate that determines the steady state pH. And here are the proteins actually in the membrane, exchanging either sodium and hydrogen or transporting, here's the sodium and hydrogen exchanger, or transporting in bicarbonate or getting rid of bicarbonate. And by that way, the cell is able to stabilize this pH at a certain level. So what did I do? I used this microscope, because remember I told you there's a dye that I'm using, and the dye is sensitive to pH. Well, this microscope has these lasers that can activate this dye at a certain wavelength. This is the emission profile of this particular dye. And you'll see that here, this is the highest peak, and it's also where these lines separate from each other. These lines represent different pHs. So here, it's really hard to tell the difference between different pHs. You can't tell the difference between a pH of 3 and a pH of 10. But here, there's a separation of the lines. And if you use this wavelength of light to examine different pHs, you can clearly see the difference between a pH of 3 and a pH of 10, because you're going to get a different amount of emission. This is actually the dye. So when you put the dye into the cell or into the solution, it goes into the cell after being acted on by an esterase, and it's trapped inside of the cell. So we're actually measuring the release of this dye as a function of pH. And this is what it looks like. Can you guys see this? Well, that is this cell. This is the neuron. And you see its processes coming off. I put this neuron in a solution that had the dye. And now all of a sudden, you can see there's a little bit of green inside of the neuron compared to out here. You actually see there's another cell here. There's an astrocyte here. Astrocytes are flatter than neurons. And so you can measure both neurons and astrocytes to see what happens when you make the cell acidic. So how do I make the cell acidic? It's this ammonium pre-post technique. If you remember your chemistry, then ammonium chloride, you'll remember, is an ionic bond. Ammonium chloride is an ionic bond. And when you put it in solution, the ammonium and the chloride ions separate and become hydrated. And now here you have this NH4, which is the ammonium. It has a charge. This ammonium dissociates into NH3, ammonia, and the hydrogen ion. And so if you're starting here at 0, which is the steady state, ammonia has no charge and can quickly go into the cell. That's the first step. This is a base, and so it causes the pH to go up. More slowly, NH4 plus rushes into the cell through cation channels like the potassium channel, and it causes a slight acidification. So that's what you're seeing here. Inside the cell, NH4 plus dissociates into ammonia and the hydrogen ion, just like it did out here. So when you take away the solution out here that the cell is being bathed in, the NH3 quickly rushes out. But you're trapping behind a bunch of acid that you just formed. That's why when you step 3, you take away the solution, it drops below the starting level. And the only way for acid to get out is through these proteins that I just described, whether it's the sodium hydrogen exchanger or these bicarbonate transporters. And what you can do is you can measure the rate of the recovery, which is phase 4, back to when it hits baseline again. Here's another phase 4. The difference between this trace right here and this trace right here is this doesn't have CO2 in it, and I've added CO2 because these transporters right here at the bottom, they are sensitive to CO2. Where is it? These transporters right here are sensitive to CO2. If no CO2 is present, you can't form bicarbonate, and so the transporters aren't working. So I'm comparing this rate of recovery to this rate of recovery. And if I take the derivative of this recovery rate, which is a change in pH as a function of time, then you'll end up with straight lines where you can actually measure the rate of the recovery as a function of pH. And you'll see that this right here, which is this rate, is slower than that rate. I mean, you can clearly see that this is moving slow to get here, and this one gets up faster. But if you actually put it into an equation, you can see there's an actual rate here. So this one is moving at 0.002 change in pHs as a function of time, where this one is moving much faster. So if you look at 6.8, you'll see that this one is moving that fast, whereas this one is moving much faster. As I said, this is older data. And the difference between this older data and what's coming out now is I have actually fully characterized what the recovery rate is in the cell. And as I wrap up, I'll tell you what that actually means. So I'm doing this in the rat hippocampus. I've loaded the dye to do the recording. This is another dye. This technique we can actually do at Virginia Union. The other technique where you're recording, we can't do that yet, but but we can show action potentials. That's not, I mean, I have equipment, personal equipment that can show action potentials, and I'm trying to get Union similar type of equipment so you guys can actually record. But you can do this right here, which is immunocytochemistry. We have incubators that can grow up the cells, if we can get the cells here. We have gas tanks where we can put different concentrations of different things and cause an acidosis to see what the cells do. We have the antibodies that can identify neurons and glial cells and cell bodies and different proteins. And so right here, I'm giving an example of what that actually means. The green ones are the neurons identified by microtubule associated protein 2. The red ones are astrocytes or neuroglial cells identified by gliofibrillary acidic protein. Because those are specific for those particular cell types, you can tell neurons from neuroglia. GAPI is a measure of, it measures the ribosomes inside of a cell, which indicates that a cell is alive. So you're looking at alive cells that are either neurons or astrocytes. And we're seeing if you really do need sodium. Because remember, I said that this is doing sodium hydrogen exchange. And if we don't have CO2, we're only looking at this particular type of thing. These ones are not active because you need CO2 to have bicarbonate. You need CO2 to have carbonate. So we're really just looking at the exchange of sodium and hydrogen. And I'm saying that you need sodium for it to recover. So to prove that, or to verify that, I did the experiment that you just saw, how to make a cell acidic. This is the recovery. And I'm doing it again, but I'm taking away the sodium. And you'll see that there is no recovery in the absence of sodium, or very little recovery in the absence of sodium. When you add sodium back, it recovers. That's both for neurons and astrocytes. This right here, which I didn't mention before, is a measure of cell integrity. If it's above a certain level, then it's considered a leaky cell, because we're measuring the dye coming out of a cell, and we don't want the membrane of the cell to be leaky, otherwise the data is unreliable. So if we say that we only want 5% of the dye to come out per minute, this is a measure of the leakiness of the cell. There's a couple of things that, as I said, are being brought out. First of all, previous studies didn't look at neurons and glial cells, or neurons and astrocytes together. This study is comparing both at the same time. Neurons are green, astrocytes are red. And you'll see that if they both are given acidosis, the astrocytes recover faster. Do it twice, astrocytes recover faster. And the recovery rate between one pulse and the second pulse is the same between astrocytes, and also the same between neurons. So this is the control data. Previous studies looked at where the neurons started, and that's how they were able to segregate the neurons. But the papers that are coming out in a couple weeks or months, however long it takes forever to write this thing, but the papers that are coming out show that using this or this before you actually do this pulse as a way to segregate the neurons and astrocytes may not be the right way to do it. This right here represents this checkpoint. So the neurons all start off here. There's a bunch of neurons. You're looking at this one, which is the green one. Here is the second checkpoint, which is right here. You'll see that after you have acid loaded the cell and look at the third checkpoint, they all have dropped to a new level. And then if you do it the fourth time, or you do it the second pulse and look at the fourth checkpoint, that level is more or less maintained. So what did I just say to you? I said that previous studies used where the experiment started off to segregate or classify neurons. I'm saying that only after you acidify them in the absence of CO2 can you rely on the data, and you should be using the third checkpoint as a way to characterize these neurons. All the data that happened that was using these ones is giving you a false impression of the distribution of the neurons in the astrocytes. It's both for the neurons, which is the green, and for the astrocytes. After you acidify them, they drop down to a new steady state. If you look at this third checkpoint, which is here, and do a histogram, you'll see that there's a normal distribution. So you can actually say, oh, the majority of these neurons are at 7.1 because that's where they want to be. And on either side of 7.1, whether it becomes more acidic or more basic, you have a decreased representation of those particular cell types. I also said that you can take this information, the recovery rate, and turn it into this. So you're seeing that it's getting, okay. So you're seeing that at a particular pH, neurons recover at a certain rate. This is a good example. At a pH of 6.9, they recover slow. As you go from 6.9 to 7.1, they recover faster. As you go a little bit more alkaline, they recover slower. So there's actually a down, up, down profile, which is what I was saying in the beginning. Proteins have a certain pH that they want to function at. If you go too acidic, that changes the structure and the function of the protein. So you're actually seeing that the function is decreasing as you become more acidic. As you go more alkaline, the function is also again decreasing as you become more acidic. And here are the actual pHs. So both for neurons and astrocytes, there's a certain pH that they want to function at. And I'm going to give a quick review and then translate it to what, the takeaway message. Just as a review, you learned about the central dogma, or it was a review of the central dogma, and that you're actually making amino acids that have properties, whether they're charged or uncharged. And acid that's either being made from metabolism or from respiration can impact the function of the protein because acid impacts the structure of the protein. Specialized neurons in the body help try to protect the body from changes in the amount of acid or changes in pH. So you have the respiratory system, and you also have the renal or kidneys to help do that at a whole body level. But inside of the cell, you have acid-base transporters that are proteins that help get rid of acid by balancing it between the extrusion and loading. That balance is the steady state. And I showed you how you can manipulate that steady state to cause the cells to become acidic and then watch their recovery. It's the difference between these cells and how they recover that determines what cells in the brain actually control breathing. And that's really what we're trying to figure out here. What are the cells in the brain controlling? And how is pH impacting their regulation of whatever they're supposed to be doing? So who can I acknowledge? First, Dr. Archer. That is the guy who I guess would be synonymous with me now because I'm at Virginia Union occupying his office, trying to fill his shoes. He had some very big shoes, and he started me off on my journey to become a neuroscientist by putting me with his advisor at Howard. And so I've started trying to do that, the same thing. Tim Brantley recently graduated and was the research technician. I put him in contact with one of the advisors that I had as a teacher when I went to Howard. And now Tim is actually in that department. So, and that guy still knows who I am. Dr. Troth, this is all networking that you're seeing right here. So he introduced me to him. Dr. Troth introduced me to Dr. Haddad, who was at Yale. No, he was at Albert Einstein. He moved his lab to California, and I went out there with him to California. Very scary because, I'm not much older than you guys as students, and I don't know what's going on. I've never been to California, but I was able to do research in his lab, and graduate, and he knew him. And Dr. Boron is actually helping Virginia Union right now because I'm here. So he is the reason that I have the study that I just showed you, the papers that are coming out in a couple of weeks or months. We're writing them right now. There's 3 papers coming up. And these are some of the students or people that I interacted with. Like, these are the people that trained me in Dr. Haddad's lab. These are the people that trained me in Dr. Troff's lab. And Austin Coley, he came and gave a talk for Journal Club before. And so the network is still there. That's actually what the Ruffin Neural Lab is. It's a networking to help minority students get into STEM professions. It's minority STEM research training. So I'm going to stop my share so that I can take any questions that you guys may have about the particular topic.
That was nice.
Thank you.
Dr. Ruffin, this is Dr. Mado. I have to run. I have a class at 1 o'clock.
Thank you very much for your time.
I have a comment and then a couple of questions. The comment is for all people like myself, was hard for me to follow your tracker, the cursor.
Oh, okay.
Yeah, if you could use a highlighter another time or a pointer.
Okay.
That will help people follow.
Thank you.
The point you're making. Question number 1. Mm-hmm. What is the source of the cells you're using?
So the previous study I showed you, they came from rats and they were in the hippocampus of rats.
Okay.
I've also done mice.
Okay. I'm glad to hear that because I lost some memory. I thought you took it from me.
And actually, some of the study, when I was studying the doing single channel recording, was done in frog oocytes. So okay. African claw frog, I would take out their eggs and inject them with cDNA and then have the protein form there so I could record from them.
Okay, the other question or comment is has anybody in your field tried to look at the COVID-19 effect on acidosis?
Because that is actually what I'm trying to look at right now. And there's somebody that will be presenting tonight in the lab about COVID and the effects of the brain. She's smiling right now.
Is that Dr. Isley?
That's the Isley over there. So I could actually upload for people that are interested in this topic, the flyer that says that she'll be presenting.
Okay.
But what does this mean? What is the takeaway? Let me make sure that I get you guys there. Your brain functions at a certain pH. Now, I showed you that diabetes and all these other things can change the level of acid inside of your body, which can actually impact your brain. Say, for example, we lived in a world where if you did have diabetes and you went to get your insulin treatment, instead of just treating you insulin to have your glucose managed, they also address the pH, which is controlling the function of the protein. If not, then you're going to continue to come back for your diabetes medicine. The cell will never be at the optimal pH for it to function. And so really what you're having here is long-term therapy for diseases that change intracellular pH. So that's instead of having the actual disease, you could actually get treated to, or maybe pH affects memory. Maybe some of the reason that we can't remember some of the things is the pH becomes so acidic that it wipes the cell clean. So as we move into this world of AI technology, where they're trying to mimic the human brain and they're trying to interact with the human brain, it's important to remember that the things that actually control the human brain are impacted by pH. And if you understand how that works, then you actually can better control the human brain. That's why it's kind of scary. But we do need representation and to be at the table when decisions like this are being made. I remember the first time I went to I remember the first time I saw a glowing brain that a student had created, because she had put a tag for YFP, or glial, not yellow fibrous protein. What does YFP stand for? It's a protein that can be attached to different amino acid sequences, and she was able to identify where all the serotonergic neurons were. So serotonin is a neurotransmitter. She was able to isolate the neurons that had serotonin in them using this different technique. And this was a student. And I'm like, wow. And then when I saw them giving mice all these different things to change their proteins, because they had mice, they were trying to basically turn into super soldiers. So just imagine you have one mouse running compared to another mouse whose proteins have been modified. The normal mouse would stop after 1 hour. The modified protein mouse would continue running for up to 4 hours. And so that's just mind-boggling that just by modifying a protein, you can modify behavior. And so it would have been good to have Dr. Webster on the line, because he is focusing on behavior. That's what psychology is all about. Before I overtalk, let me just look at these questions. Does geometry also play a role in the actions of pH? Yes, it does. So the bigger the bag, or the bigger the cell, the more liquid inside the cell, and the more stable the pH would be from changes. The flatter the cell, the less liquid. So let's make it comparable to a bathtub that has a bathtub with water in it and a cup with water in it. You add hot water, the one that has the less amount of water in it will be more affected. Whereas you pour hot water into a bath, there's so much water already there to buffer it. So that's the first question. So yes, Dr. Calitarians, pH is every cell. All cells have these proteins in them. Plant cells also. Every cell has to regulate its pH because all cells have proteins in them. And so at some level, there's a variant of these particular transporters that you could be looking at. What are the levels of wearing masks due to COVID-19? Great question. So you're actually increasing the amount of CO2 in your body. And it's going to be balanced in a couple of ways. It might change your respiration. It might change the amount of acid you're being produced through metabolism, which might cause an increase in the proteins being put into the membrane to help get it out of the cell. It also might affect your renal system. So respiratory, renal, and cellular would all be impacted. They have a thing called Kussmaul breathing, when you breathe so much that you change the amount of CO2 in your blood. Another example would be hyperventilating. You get rid of too much CO2 and it'll make you pass out. So all of these different ways the body is using to help regulate the pH. I guess I ran out of questions. Are there any more before I close this?
Yeah, I have a question, Dr. Ruffin.
Yep.
I understand that these acid-base transporters are maintaining a certain optimum pH, correct?
That's correct.
So are there any afflictions where the number of these acid-base transporters are off?
Yes. Yes. So they have actually several afflictions. You can have sudden infant death syndrome. There's also Dravet syndrome, which is an example of when the sodium channels are off. There's chronic obstructive pulmonary disease. So if you were to just, epilepsy is an example of some strokes. I mean, anytime there is a deviation from normal cell physiology, it's probably going to affect the pH. And if you have a normal cell that has the perfect number of transporters and then you start taking them away, it's going to affect how the cell balances the pH. And depending on what cell that is, it's going to affect the function of whatever you're talking about.
Thanks.
Doc, before you dismiss, I did want to just make one more announcement. We already announced that the Masters of Public Health has been approved. During this meeting, we found out that a Masters of Science and Biology program has been approved for Virginia Union. Yeah, and so they're set for spring 21. So we will hopefully try to stave that off at least as much as we can, but if not, we're going to bring it through in a quality manner. But I did want to just let the folks who are still here, I know it's 1 o'clock, so a lot of people had to leave, know that, and so you can spread the word.
Great. All right, if there aren't any more questions, we look forward to seeing you guys next week as we start having students present. Simeon, you have a question?
Yeah. I guess if the physiology of a cell is off, is there any way to use or change the pH so that the cell functions normally, even though
Yes. So I know what the pH should be. I know the things controlling the pH, and I know how to get the pH back to where it needs to be. That's why I've been going so hard, because I got something big coming. All right. Anybody else? Okay. Dr. Van, that hand is from before, right?
Yes.
Okay.
Sorry.
All right. If you guys don't have any further questions, I'm going to stop the recording. If you do have questions after this that you didn't think of, you can always contact me and I'll tell you more about the research and any opportunities that are involving neuroscience. Okay. Thank you, guys.
Thank you. Nice presentation again.